Hello, and welcome to this week's Ethical Dilemma in Science, where we're going to take a look at the concept of human organ transplantation and where those organs are going to be coming from. Now, the process of organ transplantation is based on this idea that within our bodies are going to be structures that are going to be doing specific functions that are going to keep us alive. And so we're going to have our lungs located within our chest region, which are going to be involved with helping to take oxygen out of the atmosphere and put it into our bloodstream. We're going to have a heart, which is going to be involved with pumping the blood throughout our body. Our kidneys here are going to be located within our abdominal cavity and involved with filtering waste materials out of our blood. And basically, with these structures called organs, they can wear out with the process of age, wear out with the process of disease, and basically stop functioning. And so the process of organ transplantation, a process that successfully has only been around for about 70 years. The first organ transplant was a kidney transplant in 1954. You basically go in, remove the diseased organ, and replace it with a healthy organ. Now, the science and the medicine of organ transplantation has come a long way in the last 70 years. In 2022, which is the last year in which we have data available, we're looking at an all-time high of over 42,000 transplants within the United States, 25,000 kidney transplants, and all-time records for liver, heart, and lung transplants. And so this is a technology that is providing opportunities for extended life for many individuals. So the downside of all of the advances that have been made in the science and the medicine of organ transplantation is that currently there are over 100,000 people on a waiting list in the United States waiting for an organ. And it's estimated that about every 10 minutes, somebody is going to be added onto the transplant waiting list. Overall, this means that we have a greater need for organs than we have a supply. And it's estimated that about 17 people die each day waiting for an organ transplant. There simply are not enough human organs available for the number of people that need a replacement organ. So scientists and clinicians have been looking for different ways in which they could expand the amount of organs that are available. And one of the ways that they're looking into this is the process of xenotransplantation. And so xenotransplantation is the transplantation of organs, tissues, and cells from an animal into a human being. Now, this has a great opportunity for providing additional organs, but it comes at a downside. There are risks for rejection of these animal organs by the human immune system. The human immune system is going to be fighting off and protecting the body from kind of abnormal or unusual cells that may be located, and animal cells are basically going to be viewed as abnormal cells. And so we have to come up with a way in which we can increase the amount of human cells, human organs that are going to be available that have a minimal risk of rejection by the human body. So it's within this need for additional organs for human transplantation that we ask the question, the ethical dilemma in science for this week. Should we create human-animal chimeras as a source of organs for transplantation to provide the organs that we need for those people on the waiting list? So as we dive into this question, the first thing we need to do is define what is a chimera. And so a chimera is going to be a mythical beast which is involved by the mixing and matching of various body parts from different animals. And so in mythology, this is a whole variety of things, like a Pegasus, so a horse that has wings that are allowing it to fly. In today's society, with today's science, a chimera is going to be something that basically has cells or has DNA from another source. And so mixing animal cells with human cells would be an example of creation of a chimera. So the basis of this ethical dilemma in science is an article that came out earlier in 2023 in Exploring Drug Discovery and Development, looking at chimera research, opening the doors for understanding and treating disease. And so in this case, we're looking at a mouse embryo on the left-hand side of the slide. And we've got human cells that are fluorescing, that are basically showing up as green. Another example of this over here on the right-hand side is an example of a rat-mouse chimera. And so we've got white mice here, but the brown coloring of the fur is coming from rat cells that were implanted into this mouse embryo before it started to grow. So in order to understand the science involved with human-animal chimera, basically what we're looking at is using stem cells. And so not stem cells that are isolated from embryonic sources, which have a whole variety of ethical concerns associated with it, but what we're looking at is the ability to produce what are referred to as induced pluripotent stem cells. Induced meaning that they're basically created from adult cells using different chemical factors, different chemical messengers that are going to take these adult cells and convert them into these induced, because we're creating them from human adult cells, into these pluripotent stem cells. Now, pluripotent is just a fancy scientific word saying that it has the opportunity to develop into a whole variety of other type of tissues. And so we can take these stem cells, and depending upon where we put them into an animal embryo, they could develop cells that could develop into kidneys. They could develop into blood cells, into muscle cells. They could be other cells that could develop into the pancreas and involve the hormones involved with regulating the amount of sugar that is within our blood. And then finally, we can see kind of ectoderm cells over here, cells that are going to be developing into like skin or nervous system structures. And so this slide is from Signa Aldrich, a chemical company that produces some of these factors that are going to allow us to alter these human cells into these stem cells that could be used to generate kind of replacement cells or replacement organs for an individual. So in this paper from 2018 on interspecies chimeras and current opinion of genetics and development, they described the basic science behind producing these chimeras. So in this diagram, basically what we're going to see are going to be human cells. They're going to be represented in blue, injected into the embryo or the animal at different stages of development. And so if we take these human cells and implant them into an embryo very early on in development, what we can see is the production of a whole variety of locations where we're going to have these human cells that are going to be developing. If we inject these human cells later on into the process of development, we have the potential to have specific organs. So in this case, looking at the kidneys that have been produced by these human cells. And so, as represented on this slide, we have a sheep that has basically developed human kidneys within this. And then finally, if we take these human cells and inject them into much later stages of animal development, we're going to have isolated human cells that are going to be present within the animal, but they may not necessarily form the complete organs that we need. And because we're injecting them into a more mature animal, we've got to treat them with immunosuppression to basically cause their immune system to be weakened so that they don't fight off these human cells as being kind of unusual and not normally present within them. If we inject these cells early on, the animal's immune system basically learns that they are normal cells, that they are cells that can be found normally within this body. And so we have, again, a lot of information about how we can grow these human cells within an animal model and create, either a whole variety of things or specific organs that we're going to be interested in using potentially for transplantation later on. So the theoretical process of producing these human-animal chimeras for the treatment or replacement of diseases in human beings that have damaged cells or diseased cells, tissues, or organs is illustrated on this slide. And the nice thing about using these adult cells is that we can take an individual, basically a patient that is in organ failure of some type, that needs a replacement organ. We can take those adult cells from this and induce these, treat them with chemicals, basically to produce those induced pluripotent stem cells that can develop into all kinds of different cells. We may need to alter their DNA in some way to kind of increase the likelihood that they're going to be successful, and then take those cells and inject them into an animal, either an animal embryo or an animal kind of later on in development, and then hopefully these human cells are going to be incorporated into the animal as it grows. As that animal grows up, those human cells hopefully are going to develop into an organ that is going to be available for transplantation. And so we can harvest those cells, tissues, or organs from this human-animal chimera, illustrated over here on the slide, and then transplant these back into the individual. And the great thing about this process is that we're taking cells from this individual, putting them into this process of using an animal surrogate, in essence, to grow the organ. We are going to have organs that are formed by this person's, this patient's own cells. And so that minimizes the risk of rejection, because we're basically transplanting in a replacement organ that we've created within this animal chimera. So with this understanding of the science and a appreciation for the need for additional organs for human transplantation, we get to this week's ethical dilemma in science. Should we create human-animal chimeras as a source of organs for transplantation? And below that, some specific questions. So the first question: many of the organs that can be transplanted develop from the same collection of cells within the embryo. Would it be appropriate to replace not just a small number of cells, but a larger number of cells in order to generate multiple organs from the same host, from that same animal chimera? Is there a maximum amount of human cells that could be replaced in a chimera that could still be considered as appropriate and ethical? So is replacing a few number of cells to produce a specific organ acceptable, but replacing the majority of cells, replacing the majority of the organs within this human-animal chimera with human cells, is that acceptable? Are there specific human cells, tissues, or organs that should not be created within chimeras? Are there certain types of cells that should not be used, such as nervous system cells? And so these are questions that scientists and ethicists and society needs to address as we explore this possibility of producing human organs within an animal surrogate in the use of these human-animal chimeras. So thank you for listening to this week's Ethical Dilemma in Science. Please see the description below for additional information and references, as well as a few recommended articles that will help kind of really explain the background and the science involved with this process of creation of human-animal chimeras.